In this video, we're going to discuss metabolism. This is going to be the introduction to metabolism from chapter 6. Now, what we're going to need to know. First of all, we're going to need to know examples of endergonic and exergonic reactions. We're going to need to know the key role that ATP plays in coupling, energy coupling, that enzymes work by lowering the energy of activation, that the catalytic cycle of an enzyme results in the production of a final product, and then the factors that influence enzyme activity. When we talk about chemical reactions, we're talking about having a starting molecule and then having a product and getting from A to D. However, we have intermediates like B and C that also have to be made in order to get to D. So we have a starting molecule A that needs to go through reaction to get to B, reaction to get to C, reaction to get to D. The thing about these reactions, they are all catalyzed by an enzyme for each step, a different enzyme for each step. So, a couple definitions here. Metabolism. When we talk about metabolism, we're talking about the totality of an organism's chemical reaction. So that's every chemical reaction in the organism. Metabolism manages the materials and energy resources of the cell. There are two main types of pathways. There's catabolic pathways, which release energy by breaking down complex molecules into simpler compounds. So you're going from big to small, releasing energy. And an example could be like digestive enzymes breaking down food to release energy. Then you have anabolic pathways. These are consuming energy to build complex molecules from simpler ones. So we're building up. And in this case, that would be like amino acids linking together to form muscle proteins. So you're using energy to put things together. So if we're looking at catabolic, we're talking about breaking things down to release energy. And if we're talking about anabolic pathways, we're building things up using energy. Now, what is energy? Well, first of all, energy is the capacity to do some kind of work. And there's a couple different types of energy, and you probably remember these from other classes. There's kinetic energy, which is energy associated with motion. And heat is a type of kinetic energy, which is associated with the random movement of atoms or molecules. So we're talking about thermal energy, we're talking about the movement of molecules. Then there's potential energy, which is stored energy as a result of its position or structure. So many times you'd think of the energy that's found in the bonds. Now, energy can be converted from one form to another. So we get in the idea of chemical energy being converted to mechanical energy, which then gets converted into electrical energy. So we are transforming the energy from one form to another, or converting it. Now what we have here is we have a diver that's on a platform up here, and then we have the diver that has already taken the dive and now is in the water. So we have the potential energy up here on the platform, we have the kinetic energy as the diver is diving, potential is being turned into kinetic energy, and then the diver at the bottom has less potential energy than they did on the platform. Now, when we're talking about energy transfers and transformations, we're talking about thermodynamics. And in thermodynamics, we're talking about a couple different systems. A closed system in which you have like a liquid in a thermos is isolated from its surroundings. So it's closed. Nothing is coming in and nothing is going out. However, when we're talking about organisms, we're really talking about open systems, and where energy and matter is being transferred between the system and its surroundings. So there's this flow that is happening between them. And organisms, again, are open systems. We have food coming in, we have wastes going out, we have energy flowing through us. An example of a closed system would be this picture here, where we have a higher level of water over here. So we have potential energy here. We have kinetic energy going through the water wheel, and it's lighting a light bulb. This is like water flowing through a dam, turning a turbine, and flowing into the capture chamber here, but it is less energy. The problem is, as it's flowing, it all reaches the same level. And when it reaches the same level, it can't turn the water wheel anymore. And so in that case, the light bulb goes out. So this is a closed system. Now in an open system, we have the materials are going through multiple light bulbs, and we're gradually reducing the amount of energy that is available to do work because it's being used to light these light bulbs, and it's moving from a higher potential energy to a lower potential energy in the process of transferring the potential to kinetic to light those light bulbs. And you can think of this as food coming into the organism, waste going out of the organism, and in the process keeping the organism alive. We talk about the first law of thermodynamics. We're talking about that the energy in the universe is constant, and the energy can be transferred and transformed, but it cannot be created or destroyed. So that's the first law, and you guys have talked about that in the past. This is also called the conservation of energy. So the first law of thermodynamics states that we can transfer energy from, for example, this fish, that is chemical energy, into this bear, but it's not being created, it's not being destroyed, it is just being transformed. Well, the second law of thermodynamics states that every energy transfer or transformation increases the entropy or disorder of the universe. So we're increasing the chaos within the universe, the lack of organization, the lack of order, the lack of usable energy. So during every energy transfer or transformation, some energy is unusable and is lost as heat. So nothing is 100% efficient. We have even machines that we have created, they have a level of efficiency, but nothing is 100%, where you turn the energy that you're putting into it into the actual the work that's coming out of it. You don't get 100% transfer of energy. So there's always going to be some lost as heat. So even though the bear ate this fish, and it's turning it into muscle work, and it's running, and it's giving off a little bit of carbon dioxide and water, and that's kind of those waste products, but you're also losing this as heat. Now, free energy. There's some type of energy called free energy, which is a part of the system's energy that's available to perform work. And when we talk about free energy, there's something called delta G, which is a change in free energy. So we go from a certain amount of free energy to a new amount of free energy, and depending on what that change is, that tells us something, what has happened. So what we can do is we can take the delta G is equal to the final state minus the initial state. So final minus initial. And in exergonic reactions, energy is released. We consider that a spontaneous reaction, and delta G is going to be less than 0, because if the final state where energy has been released is less than the initial state, then this minus this is going to give us a negative number, and that's where the delta G is going to be negative. Endergonic reactions are opposite of that, and that's energy is required, and it absorbs the energy from the surroundings, and the delta G is going to be greater than 0. The final state of the compounds, the molecules, are going to be greater than the initial state. Therefore, this minus this is going to give us a positive number. So exergonic reactions are going to be negative delta Gs. Endergonic reactions are going to be positive delta Gs. Exergonic, exiting energy is released. Endergonic energy, end meaning in, energy is being input into the system. We can look at a couple different graphs that show us what an exergonic and endergonic reaction would look like. And exergonic is where you have the reactants at a higher free energy than the products. And so energy was released. Therefore, this would be a negative delta G. If we look at an endergonic reaction, we can see that the reactants have less energy than the products, or you could say the products have more energy than the reactants. Therefore, energy went in, and that is a positive delta G. When we talk about work, the cell does 3 main kinds of work. There's mechanical work, there's transport work, and then there's chemical work. Now, cells do a great job using these energy resources to do the work by energy coupling. They use an exergonic process to drive an endergonic process. So energy is released to then power the endergonic reaction where energy is needed. So energy exits exergonic process and enters the endergonic process. ATP is a cell's main energy source in energy coupling. And an ATP is an adenine plus a ribose, also called adenosine, and then 3 phosphates. That's where the triphosphate comes from. So here's our adenine and our ribose. Together we have the adenosine right here. And then we have 1, 2, 3 phosphates. So that's ATP. When the bonds between the phosphate groups are broken by hydrolysis, if we remember this back when we were talking about polymerization, monomers becoming polymers or polymers becoming monomers, hydrolysis energy is released. And this release of energy comes from the chemical change to a state of lower free energy, not in the actual bonds themselves. So as we break off this phosphate that was attached to the ATP, we're left with a DP plus this phosphate and a lower amount of free energy here, and that energy is going to be transferred into doing some sort of work later on. How does it work? The exergonic release of phosphate is used to do the endergonic work of a cell. So as the energy exits the ATP, it is going to enter the endergonic work of that reaction. So when ATP is hydrolyzed, it becomes a DP. That's adenosine diphosphate. So energy from catabolism powers the anabolism. The energy from exergonic reactions powers the endergonic reactions. So here's a picture of some of the things that can happen. We talked about motor proteins in the past as ATP can help move vesicles across a microtubule. We could talk about, in this case, we have the sodium-potassium pump. Here's ATP powering the sodium-potassium pump so it can exchange those materials. But we also have down here reactions where you can have amino acids that are being combined into a different amino acid, and in the process is being powered by ATP. So that's mechanical energy, this is transport energy, and this is chemical energy. Now, what is a catalyst? Well, a catalyst is any substance that can change the rate of a reaction without being altered in the process. An enzyme is a specific type of catalyst called a biological catalyst. Now, what do? Well, they speed up metabolic reactions by lowering the activation energy, which is the energy needed to start the reaction. So, for example, we have sucrose over here, and we have water because we're doing on a hydrolysis. We're going to use water to break things apart. If we add sucrase, and you'll notice that its ending is ase, which this ending is ose, that means sugar, and this ending ase means enzyme. So sucrase is going to assist in the breaking down of sucrose, the hydrolysis of sucrose, into its component parts, which is glucose and fructose, and that is going to be done by adding the hydroxyl group over here and the hydrogen over there. But it is done by lowering the amount of activation energy necessary to get this reaction started. So when we talk about activation energy, what we're talking about is that all reactants going to product need some sort of energy to get them over the hump, to kind of take those bonds that are holding the molecules together, the atoms together in the reactants, and make them enter some transition state where they're a little bit wiggly and easily able to fall apart. Well, that energy is called the activation energy. And we can see that this black line is the activation energy necessary without an enzyme. So you need more activation energy without an enzyme to make this happen. Now, this red line is the activation energy necessary if you are using an enzyme. So you can see we have less activation energy here than we do on this black line. Now, if you have less energy necessary, then it is also going to actually speed up the chemical reaction. Because if you have less activation energy necessary, then you won't need as much, and you'll be able to get to the product much quicker. When we talk about enzymes, we talk about another thing called substrates. Now, substrates are what the enzyme is going to work on. It's kind of like the reactants of the enzyme. They are very specific in what they're going to fit into in terms of the enzyme, because all enzymes have a specific shape and they fit a certain substrate. And every substrate fits specifically into a certain enzyme. And so the reactant that an enzyme acts on is called the enzyme substrate. And the enzyme binds to its substrate, forming an enzyme substrate complex. The active site is the region on the enzyme where the substrate binds. So here is the enzyme, here is the active site where a substrate will bind to. So looking at these pictures here, we have enzyme, active site, we have enzyme, substrates have entered the active site, and we have two substrates, and then the enzyme works on it, and then the products are going to come out. And you can see that we have this green and orange, and then they become a blue and a yellow. You'll also notice the enzyme has not been changed in any way. So as the substrates enter, they get worked on, they get modified, they get turned into product, and then they exit, and the enzyme is ready to go again. Another example would be of enzyme action is specifically talking about catabolism. So here's an enzyme, here is the substrate, here's the active site, here is the enzyme substrate complex together, and here is what's the enzyme plus product. And we can see that this was a catabolic process because the substrate, one thing, was broken down into two things. So that would be catabolism, and energy was released. Now, if we're talking about anabolism, we're talking about things coming together, building up. So we have the enzyme, we have the two substrates that are entering the active site. The active site works on the two substrates, puts them together, and so we have an enzyme and then one much larger product at the end. That would be an anabolic reaction. When we talk about enzymes and their substrates and active site, what happens is the substrate fits into the active site, and you can actually see the enzyme changing its shape. It kind of gives it a big squeeze or like a hug or a handshake. And that handshake, you can see that it's more loose here and very tight here. That handshake or tightening up actually assists in the destabilizing the bonds within that reactant. Now, enzymes can be affected by a couple different things. We're going to focus on temperature and pH. And if we look at this graph here, we can see that the optimal temperature for a typical human enzyme is 37 degrees Celsius. So here is a typical human enzyme, 37 degrees Celsius. That is the normal body temperature. The normal body temperature is 98.6 Fahrenheit, but it's actually 37 degrees Celsius. So all of our enzymes work best at 37 degrees Celsius. But if you were to look at a heat-tolerant bacteria, a thermophilic, heat-loving bacteria, you would find that they have a much higher tolerance for temperature. This one happens to be 77 degrees for its optimal temperature. Down here, you can see that we have some optimal pHs for different enzymes. This pH is at 2, which is very acidic, and that's for pepsin, which is an enzyme that breaks down proteins inside your stomach. Stomach environment is very acidic because it needs to be in order for pepsin to work and break down those proteins. But we have a different enzyme called trypsin that is found in the intestines, and that's at a pH of 8. And so the pH of 8 is basic, and so when you go from your small intestine to your, from your stomach to your small intestine, you have a change in pH because you have enzymes that work at different optimal pHs. Now, when you go away from the optimal pH, the enzymes actually change shape and become denatured. Remember that proteins have 4 major structures. They have the primary, secondary, tertiary, and quaternary structures. Well, when you start changing the pH, you start moving away from the optimal pH, or when you increase it past the optimal temperature, then you start to denature these proteins, denature these enzymes, and they can't work as well. And when they can't work as well, you end up having change in shape and therefore loss of function. There are also some things called cofactors that are going to be helping them out. Factors, they're non-protein enzyme helpers, such as minerals like zinc and iron and copper, the metals that we find in our diet. When we talk about coenzymes, those are organic factors, and those would be vitamins. And so all of your vitamins, vitamin A, B, C, D, E, K, et cetera, those are coenzymes. Those are organic. Cofactors are non-organic, non-proteins, but they are both important in order to make enzymes function. Enzymes can also be inhibited or stopped from working by a couple different things that are actual molecules that interfere with its function, not just like temperature or pH. A competitive inhibitor will bind to the active site, competing with a substrate, and gets in the way of the substrate. A non-competitive inhibitor binds to another part of the enzyme, but it does change the enzyme shape and creates an active site that is non-functional. So let's make sure we understand these two terms here. Competitive competes for the active site, competes with substrates. So it's another molecule that kind of fits in the same spot. A non-competitive inhibitor does not compete with the substrate, does not compete for the active site, but instead binds somewhere else. And when it does, it changes the shape so that the enzyme is non-functional. So here's an example of inhibition. And here's the enzyme, here's the active site, here's the substrate. That's supposed to fit in there. Here's an enzyme, here's a competitive inhibitor getting in the way of this substrate so it can't bind, so it can't go from reactant to product or substrate to product. And here is a non-competitive inhibitor binding in a place away from the active site that is changing the enzyme shape and not allowing the substrate to bind here. Now, there's various ways you can regulate enzyme activity. You could regulate the metabolic pathways. The cell switches on and off genes that can encode specific enzymes. So if you're going to regulate enzyme activity, you could basically produce more enzyme or produce less enzyme based upon what the genes are doing. Because remember, genes are responsible for making proteins, and those proteins are enzymes, and that's how they function. But you can also have what's called allosteric regulation. You can have proteins functioning at one site that is affected by the binding of some other molecule at a separate site called the allosteric site. That's almost like that whole concept of non-competitive inhibition, but this time we're talking about a molecule binding at a site that is away. We're talking about a molecule binding at a site, a separate site called an allosteric site. And you can either have an activator or inhibitor. And so an activator stabilizes an active form of the enzyme, and an inhibitor stabilizes the inactive form of the enzyme. You can also have something called the cooperativity, where one substrate actually triggers the shape to change so that the other active sites increase their catalytic activity. So in this picture here, we have an allosteric site. And what's interesting about these, they're oscillating back and forth between an active and an inactive form, active and inactive, active and inactive. Well, at some point in time, a molecule shows up that's an activator, and it gets stuck into a position called an allosteric site that keeps it in a stable active form. And so you can see that these little spots here are going to be ready for substrate. These would be like the active sites. So it's ready to do its work. But in this form, if an inhibitor is present, then it ends up getting stuck in the off position after it's oscillating back and forth. And as it's turned into the off position, you end up having all of the active sites that are shut down. And so now it's off in a sense. So this is locked into an on position, and this is locked into an off position. So it's an activated and inhibited situation. So that would be an activator, and that's an inhibitor. We also have what's called cooperativity, where you have substrates that become the activator. So you have activation, this oscillation from an inactive to active, inactive to active, inactive to active. But at some point in time in the active form, you had a substrate that has shown up on the scene and locks it into its active position. And that one substrate allows for more other substrates to bind in order to be worked on by that enzyme. Now, the last thing we're going to talk about here is what's called feedback inhibition. And at some point, when we're done getting our product, we want it to turn the machine off. So the end product of a metabolic pathway shuts down a pathway by binding to the allosteric site of an enzyme. Why would we want this to happen? This is going to prevent wasting of resources, and it also increases the efficiency of the cell. In this picture, we can see that, again, we're back to where we started. A turning into B, turning into C, turning into D. And in this case, our final product is after it. So we start with an initial product and we're going all the way through these chemical reactions, this metabolic pathway in order to get to our end product. So the big idea here is that we're going to take threonine and try to turn it into isoleucine. The way we do it is we have to go through 1, 2, 3, 4 intermediates to get there. And to get it to 4 intermediates and finally the final product, we need 1, 2, 3, 4, 5 enzymes to make that happen. As our enzyme becomes available and our initial substrate bonds, it turns it into the necessary intermediates and finally get our end product. But we don't want endless end product. So to turn the machine off, it turns out that we can have a feedback mechanism where the end product feeds back to an allosteric site, a site away from the active site, and locks in and changes the shape of the active site, because you can think of this as a non-competitive inhibitor. And when it turns this shape, when it creates a shape change here, that then prevents the substrate from binding in there. And if the substrate can't bind in there, you're not going to get any more end product. That's called feedback inhibition, where the product feeds back to turn off the enzyme at a site away from the active site to change the active site so that the initial substrate can't bind, and that turns off. And that was a brief overview of Chapter 6, Introduction to Metabolism.